Dr. Nanja van Geel. She is a professor of dermatology at Ghent University from Belgium. And I would like to give her a new name, which is the Queen of Vitiligo scores, as she is very interested in making scores for evaluation of vitiligo, which really changed the way how we evaluate the severity of the disease, especially on research. And she's going to give us her presentation about an international collaborative project to standardize the assessment of vitiligo during research. So you are welcome to join, Nanja.
Good morning, or good evening, or good afternoon, of course. So I would also like to thank Samia, my colleague, for the nice introduction, but also the co-chairs for this great event. This is really wonderful to have us all together and to share all our research. But also the team behind the co-chairs, of course, including the technical support, in particular, Dr. Patrick Dawkins. I think this is really nice, organized. So I will now try to share my screen. Yeah.
Yes, it's ready.
Okay. So I'm pleased to share with you the protocol, a new study actually to standardize the assessment and monitoring of vitiligo in trials, registries, and clinical practice. So we have the intention to develop an international agreed minimum data set or outcome set for vitiligo. And just a second, I have a technical problem here. Okay, so the consensus is really important to achieve, as this will support the selection of consistent outcome measures and instruments during the setup of new trials. This consensus is also important to enable the comparison and pooling of outcomes between institutes, researchers, and trials. It will also ensure that the findings are relevant to clinicians and patients. So the possibility to compare and to pool our outcomes is actually a key element in doing research. It will also give us more insight in the management of vitiligo and the understanding of the disease and to estimate prognosis. It will also help us in the development and evaluation of therapeutic options and for pharmacoeconomic evaluations. It will also help us to define evidence-based guidelines. So we would like to define what and how to measure, and this for 3 settings: for clinical trials, for registries, and clinical practice. This project will consist of 2 parts. We will first focus on the construction of a minimum data set. And in a second step, we will try to get an international approval, a consensus. The 3 data sets may differ in the number of items they will include. They will include physician and patient-reported outcomes, as well as contextual factors. And in addition, they will be supplemented by consensus on how and when to measure. So we will focus on measurement instruments and the timing of assessment. For this project, we set up a collaborative network, including special interest groups for vitiligo, but also patients' organizations. And for the methodological support, for instance, an organization like COSMEN. This project started in 2019. And more recently, the discussions were also performed within the IDEUM group. In the meantime, many conference calls were performed and we developed a protocol. It might be important for you to know that a relevant research has been done already related to core outcome sets, in particular for trials. So this was published in 2015. The essential core outcome sets were defined, including repigmentation, side effect, and maintenance of gained repigmentation, as well as the recommended core outcome sets. Also, recommendations for target lesions in randomized controlled trials were published. So what is now the difference between this previous core outcome set and the minimum data set we would like to create? The previous core outcome set did not include demographic data, details, diagnostic data, and also treatment-related variables. The minimum data set will include the contextual factors in addition to the outcomes. And for this project, we will also look for registries and clinical practice in addition to trials. So how to measure? Some work has been done in the past. If we look to 2012, there was a systematic review. In this systematic review, they evaluated the quality of the instruments. So they looked to the validity, the reliability, and the responsiveness based on the Cosmin checklist. However, based on this systematic review, none of the available measurement instruments could be highly recommended for use. So, and this is also why the home roadmap suggests to develop or validate new instruments if existing instruments are found to be inadequate. And this was also seen after 2012. Many new tools were developed in the previous years to fill the gaps. So the new instruments did not only include assessment instruments for impact, but also for extent or global assessments, and also new evidence for measurement properties of existing instruments was provided, for instance, for the VIS. So coming now to the project, the methods and design. So what do we hope to achieve? We would like to construct a minimum data set based on 3 settings, and subsequently would like to get this approval based on nominal group technique, electronic survey, so the eDelphi procedure, and a consensus meeting. In addition, we would like to evaluate the quality of current available measurement instruments and to reveal their possible needs. This will be done based on a systematic review and consensus meetings. So we would like to create 3 panels for each setting, and the panel members will be selected from different continents. The panels will consist of vitiligo experts and clinicians, but also by patients, and we also will include other stakeholders. We would like to include 30 to 35 members per panel. This is based on feasibility aspects and also limitations we experienced in the past. So for the methodological guidance, we will rely on the home roadmap, as well as on the Cosmin and AMRACT filter. They can be used for the quality assessment of the instruments. This project will consist of 10 steps. Within the first step, this is actually the current stage, we would like to define the scope and applicability for each setting. We would also like to finalize a protocol and to define the panel members. Within step 3 to 5, we will focus on the construction of the maximum data set. First, the first version will be created based on a systematic review, based on an update of the systematic review, as well as based on variables in existing clinical registries and expert opinions. A second version will subsequently be created by the panel members. They will add missing relevant variables based on nominal group technique. For steps 6 and 7, we would like to construct a minimum, so the reduced data set for the 3 settings. And this will be done by each panel separately. We will rely on ranking and voting rounds until agreement will be achieved based on eDelphi procedures. We will use a predefined methodology for this step. For step 8, we will focus on the consensus on how and when to measure the items included in the minimum data set. For this, we will rely on the systematic review and a consensus meeting or meetings. We will evaluate the quality of the instrument. We will look to the measurement properties, and we will use for this the Cosmin criteria as well as the Amrecht filter, which is quite similar. So we will look to the validity, reliability, and sensitivity to change of the different instruments. We will also look to the feasibility of, aspects of the instruments. We would like to answer 2 questions. Are the existing measurement instruments adequate and suitable for each domain? And is there sufficient evidence for their quality, so for the measurement properties? We will, if this is required, complete validation studies for currently available measurement instruments, or if this is necessary, we can consider to develop a new instrument for a specific domain. Within step 9 to 10, we will look for a global approval, and this will be done in a large group of participants, including all the panel members, but also other experts, collaborators, and stakeholders. We will, within the meetings, present the minimum data sets. We will discuss them. We will explain why we included or excluded some items. We will evaluate this together with all the members of the team. We will also pay specific attention to the relevance, applicability, and feasibility aspects of the datasets. And within a final meeting, we will solve remaining issues and get finally a global approval. And of course, for the future, we would like to disseminate and implement our datasets by the use of publications, presentations. But also we would like to provide guidance material, including training manuals on the instruments or a template for the uniform collection and the reporting of the data. We would also like to evaluate the use of the data sets within different centers or within existing database to detect shortcomings. And subsequently, we can still optimize the data sets. And of course, we would like that the data sets can be integrated into national and international collaborative efforts, and that they will be available for use in trials and daily practice. However, we should keep in mind that this remains a dynamic process. And if indicated, so if new evidence is available, the data sets needs to be revised and modified. I thank you for your attention.
Thank you, Nanja. That was a wonderful talk. And I think Samuel will be joining us in a second, but we're going to go ahead and start.
I'm sorry, I was muted. I was muted. Yes.
That's okay.
All right.
All right,
yes,
I'll pass it over to you, Samia.
All right. I don't have questions at the chat box yet, but I have a few questions for you. How long do you expect this project to stay? I mean, how long do you think it will take time? I mean, I think there is a lot of work, and maybe if we reach step 9, step 10, would this be 2 years after or 3 years? I mean, did you plan for the time?
Yeah, this is a good question because we also consider consider to start with one setting first, and then to use the technique and the information for the next steps, for the two other settings. But it will also depend on the possibilities of the collaborative network of funding we will achieve. So I think it's not exactly known yet, but we hope to get that, to have it done, or maybe to have a huge step done in 2 years. I think this is the aim. But of course, you know, it's a dynamic process. It's a collaboration, so you always need more time if you have many people involved. So I think we need to be realistic, and it will be something for more than 1 or 2 years, probably. Yeah.
Yes, but I mean, we need to, I think before you start working or whatever the group is, determining the time period makes the work goes faster.
Of course.
Yes, of course.
You're right.
Yes, and about your lecture yesterday, it was about the severity and the stratification of the severity. I mean, I sent you a question, but maybe it was late. How would this stratification help us for the clinical management of the patients?
Yes, I think it's already very important if you refer to the definition of, for instance, mild, moderate, and severe. This is one of your questions. I think it's related to stratification technique. This is a good question because this was actually the reason why I was interested in this question. Because if you look to trials, they want to include moderate or just severe cases, for instance, but not know what is a severe case in vitiligo. So if you look to other examples like psoriasis, you can see that all the research is defined based on the inclusion criteria of severe or very severe psoriasis cases. We do not have that yet. So I think this was actually one of the reasons that we were interested in this topic. Because we need to know what is mild, what is moderate, what is severe. So it's not only relevant for inclusion, but also for reimbursement criteria that maybe will follow in the future. And of course, also for your management, I think we need to define guidelines. For this, again, you need definitions. You need definitions of the specific categories that we mentioned. So yes, this was actually the basis. So your question was,
I mean, if we want to classify the severity, I mean, this classification needs to be attached to the management. So, so that when I say the management plan, I will use this classification, so it will be perfect.
No, I think it can help us to define the management, because it's really, yes, exactly.
All right, I thank you very much for the nice presentation and for your efforts all the time to make the assessment of vitiligo more easier and clearer.
Thank you.
So, yes, I would like to present our next speaker.